Thanks, Sean. And thank all of you for coming. So my presentation tonight is part of the Visualizing Biological Data workshop, but it's also the Science and Society lecture, as Sean mentioned. And that's part of an ongoing forum to promote awareness of the impact that the life sciences have on society. Most of you are here from the workshop, but some of you are here because you might be interested in this interface between research institution and the public at large, or you might be interested in seeing images used to convey meaning. So what I want to do tonight is show you how I express scientific concept visually to convey information for artistic expression and to participate in this process of research through data visualization. This shorthand represents one of the most celebrated molecules in biology. It's simply shown as aligned with the two ends labeled differently. And I think many of you in the scientific community would recognize this molecule. Actually, with a show of hand, how many people know what this represents? Okay, so it's a well-known visual convention that represents DNA. And those familiar with DNA knows that it typically exists as a pair, and those pairs run in opposite orientation. And they're held together by chemical bases, and this whole thing is twisted up into a double helix so that the periodicity is about 10 of those bases per turn. And these strands are not evenly spaced so that when they're twisted, they form these things called major and minor groove. So that's a lot of information behind a very economical representation of DNA. On the right, most of those physical features are spelled out. And these two things form a continuum of representation. And their purpose, obviously, is very different. And I think to create effective visual communication, it's really trying to hit the right spot in that continuum that matches the purpose of the communications and also the audience that you're targeting. But this continuum also represents things like language. Do you use more direct, literal language, or you use more metaphorical language? And also things like accuracy. Do you go for scientific reality, or do you focus on the salient points to get the message across? This idea of picturing science comes from a long tradition of drawing and documenting science for the purpose of communication. And this tradition goes back to people like Da Vinci, Haeckel, Dürer, and others. Da Vinci was renowned as a painter, and as a scientist, he really advanced the state of anatomy and medicine with his study, writing, and of course, illustrations. This is Max Bredel. He's sometimes referred to as the father of medical illustration. He's a German artist who, in 1894, came to Johns Hopkins to work with surgeons to illustrate pioneering surgical techniques. And on the right is one of his techniques called carbon dust that he's well known for. In 1911, he established the first graduate program in medical illustration at Johns Hopkins. And I'm a graduate of that program, and this was my first drawing assignment. It's of a human hip bone, and it's done in this age-old technique called carbon dust. And in this technique, you take carbon pencils and you grind it down to a powder, a really fine powder, and then you apply this powder with a paintbrush. And then the way that you capture this detail is you go back with pencils and then erasing back to the white of the paper. And at the end of every session is a complete mess, and it looks like you've just come from the coal mine. And it was the first and probably the last time I'll ever use that technique. So more commonly these days, though, people use computer and software like Photoshop to aid their work. And this is another drawing of mine during that time. It shows how the blood flows around the elbow so that when you bend it, blood still gets around. On the left is a colored pencil sketch. And then on the right is a painting that I did behind that sketch in Photoshop. One of the subspecialty of medical illustration is called surgical illustration. And in surgical illustration, I learned about this idea called a surgical moment. And a surgical moment is a pivotal step in a surgery where all of the relevant anatomies have been identified. They've either been protected or they're prepared for some next step. And it's pretty tense in the OR until this moment is reached. And there could be several of these moments. I'm showing you the work of my teacher. It's done in watercolor. And this is a procedure to treat pancreatic cancer. And what you're seeing here is that the intestine's been cut, and the pancreas has been divided. The branches of the arteries and veins have been tied off. The duct leading to the gallbladder has been exposed. So medical illustrators are trained to capture these moments because these illustrations are the visual landmarks that surgeons communicate their technique, and it's a way that they learn. This is a later step in that same surgery. So the head of the pancreas is lowered into and ligated to the intestine. And this pair of illustration clearly describes that process. And you can easily imagine what happens when you pull on that set of sutures and see this thing drop down. The reason that illustration endures is that it allows the artist the ability to edit the scene. With imaging techniques like photography, you basically get what you see, no more, no less. With illustration, you can even show what can't be seen. So in my current position as creative director at the Broad and principal in my own design firm, I work with researchers and people from many other fields to translate science to many audiences. And my work these days don't resemble that of the medical illustrator in content, but I think the core approach to putting together illustration, whether it's medical or scientific or information graphics, is still very similar. I think it's a process of sorting through lots of information for what's relevant, and relevant as it relates to the message and the purpose of that illustration, and then relying on visual cues like colors and composition to put a figure together. One of those audiences is the scientific audience, and I think scientists, they care about how experiments are done and the reproducibility of results. And so I think overview figures like this one goes a long way to providing that roadmap for understanding experimental design and for understanding the data that comes out of it. And of course, figures are really the mainstay of scientific communication. They aim to convey a very specific body of information. In turn, I take this primary literature and explain it to a NOVA audience. And NOVA makes films and documentaries about science. And translating this work to this audience requires me to shift myself on that continuum, coming up with new representation instead of using old conventions and using more metaphorical language and less direct literal language for it. I've worked on a number of these segments in the last 2 seasons. This one's called The Marathon Mouse, and it's about how these researchers have discovered 2 drugs that mimic the effect of exercise, not commercially available yet. I think in addition to the audience, I think the media really affects how you present the information as well. And I think for TV and for web, that presents its own challenges. The television program tends to use very colorful metaphors to explain science at a high level. Gas tank running low, mitochondria is represented as a factory, churning out energy, and so forth. Sort of the hard science, though, it's reserved for the web. And I think the web is a very good place to show complex information because I think you can arrange it in this 3D matrix so that the user can scan across horizontally on a lot of different topics and then dive deep when they can and they need to for additional information. I want to show you this illustration from the New York Times to highlight an important point about scientific illustration, which is scientific accuracy. The audience in the New York Times, at least the ones that read science news, I think are looking for major concepts. And this one is about how stem cells control which genes are turned on or turned off during development. So when this figure came out, that very morning I got this email from a very prominent scientist, and it reads, "Saw your diagram in the Science Times. Very nice, but oops, a bit wrong. DNA wraps 1.67 times around the nucleosome. Your diagram shows more than 2. But what he didn't notice is that the DNA is also wrapped around the histone in the wrong direction. And now I know why this building is up in the hills, because it doesn't want to get caught like I did. But this snafu, I don't think compromised the intent of the illustration, which is it has to do with the pattern of these chemical modifications on the histone tails. But what it does do is undermine the credibility of the whole piece. Another thing that came out of this was, I talked to you about showing about conventions at the beginning. And there's also conventions in the media, and it has to do with the representation of chromosomes here. When I sent this sketch in, this pencil sketch in, I showed the chromosome as a single bar. And the New York Times illustrator turned it to this very familiar X shape. And I think this X shape poses some problems. Let me explain. So we normally get one chromosome from mom and one chromosome from dad. And along with it, we get mom's variation of the gene and dad's variation of the gene. And so when the cell gets ready to divide, it duplicates the DNA. And at this sort of very fleeting moment in metaphase, there's actually 4 copies of everything. And I think this confuses the idea that you get, the idea of alleles, that you get 1 copy from mom and 1 copy from dad. And it also confuses this idea of how the genome is organized into pairs of chromosomes. And this pairing is this thing here, not the X. I was asked to design a cover to commemorate the completion of the dog genome sequence. And I find these conceptual pieces much more challenging to do than those figures that I showed you previously. Because in a lot of ways, creating a figure is very straightforward. All the information is there. It may be jumbled up in this bucket, but it's a matter of sort of pouring out all the information and sorting it into a hierarchy, primary information, secondary information, and extraneous information. But there's a lot of cues that you can use to guide yourself through this process. You know, if it's a comparison, then you emphasize what's the same and what's different. You know, if it's a process, then you show visual cues from one step to another so somebody can follow that. But in the design of a cover, there's a lot of things you can do. And in this case, the dog genome was very scientifically significant because dogs get many of the same diseases that humans do, and some breeds are more prone to certain types of diseases. And this fact, combined with the careful records that breeders have kept over the years, makes the dog genome a treasure trove for looking for the genetic roots of certain diseases. So in designing a cover like this, the obvious starting elements, of course, is the dog, and then some representation of DNA. Right? And this was 2005, and people are still celebrating the 50th anniversary of the discovery of the structure of DNA. And so I very much wanted to use this photo that's become iconic to symbolize the discovery of DNA and this celebrated double helix shape of the molecule. But I needed to add the dog in here somewhere, and I felt like it was a sacrilege to really tamper with this photo. But with permission from James Watson and lots of cajoling from the Nature editors, we put Tasha the dog in here, whose DNA was sequenced.